In the next video, we're going to talk about what's called the link reaction and the Krebs cycle. Now, the last part of glycolysis involved the production of pyruvate, and that happened outside of the mitochondria, inside the cytoplasm. So this would be in the cytoplasm. And pyruvate, if you remember, is a 3-carbon molecule, and for every glucose you make 2 pyruvate molecules. Now, pyruvate will make its way into the mitochondria. Now remember, the mitochondria is a endosymbiotic, which is a fancy word for not belonging to your cells, but was originally some kind of microorganism, and it contains all of the molecules that do the rest of, or the majority of respiration. Now, pyruvate can enter through the outer membrane of the mitochondria easily because it's completely permeable, but the inner membrane is completely impermeable, and so it will enter the cell, the inner membrane, or the matrix of the mitochondria through the inner membrane, in a process known as the link reaction. Now, what's produced is something called acetyl-coA. Now, acetyl-coA only has 2 carbons, which means that link reaction must produce a CO2. And because we produce a CO2, we also produce NADH+ from NAD. So a reduction reaction has occurred, electrons have been accepted, and that's really important because we're going to make a note of these NADs as we go through. So we now have acetyl-CoA. Now acetyl-CoA has 2 carbons, and it will be combined with a molecule called oxaloacetate. And oxaloacetate has 4 carbons. So the product will have 6 carbons and is called citrate. Now, you don't need to know the names of any of these steps that go, but every single step is referred to as an intermediate. So the first step involves the loss of a carbon dioxide, and it forms a 5-carbon intermediate, which means you produce CO2. You also produce an NADH+ from NAD. So this is reduced NAD. The next step involves going to a 4-carbon intermediate. And again, you produce a CO2. And you make a reduced NAD. So at this point, you're just rearranging chairs, and you go from 4-carbon to 4-carbon, eventually ending up back at oxaloacetate. Now, at some point, you actually make an ATP from ADP. This is what's called substrate level phosphorylation, and that's a really fancy term, so let's write that down. So substrate level phosphorylation. And all that means is I've nicked a phosphate from something and I've given it to something else. So in addition to this ATP, I'm going to make reduced FAD. So this is a new molecule we haven't seen before, and it makes FADH2. FAD is a bit like NAD. It's an electron carrier. FAD stands for flavin adenine dinucleotide. And again, it's just an electron carrier. Now, unlike NAD, it's not mobile, but that doesn't matter. We're going to learn more about what FAD does later. So this is the Krebs cycle. And Krebs cycle is just a process of a series of oxidation and reduction reactions where we're producing electrons that are being captured by either NADH or FADH. We also make one over here. And these NAD will go off to the electron transport chain. So these are all going to go off to the next step, which is the electron transport chain. And we'll talk more about that later. So to go through that again, two pyruvate are made from each molecule at the end of glycolysis. So up here, this was glycolysis, and it happens in the cytoplasm. And that's the last thing that happens in the cytoplasm in aerobic respiration, because everything else happens in the mitochondria. So this is my very badly drawn mitochondria, but pyruvate can enter through the outer membrane completely happily, because the outer membrane is permeable, but the inner membrane is impermeable, and so it gets taken up in the link reaction, which is a process that forms acetyl-coA. And in doing so, because acetyl-coA has 2 carbons, pyruvate had 3 carbons, a CO2 is made. In addition, because that's a redox reaction, we make an NADH, which is going to go off later to the electron transport chain. Now that acetyl-coA is going to get combined with oxaloacetate. So 4-carbon oxaloacetate will combine with 2-carbon acetyl-coA, and citrate is made, which has 6 carbons. Now, some people will say, what happened to the CoA? The CoA gets recycled and used again later. Once I've got citrate, which has 6 carbons, I do a number of redox reactions. So the first thing is to change it into a 5-carbon intermediate. Again, a CO2 is removed and an NADH is made because it's a redox reaction. That 5-carbon gets turned into a 4-carbon. Again, we lose a carbon as carbon, and we make an NADH plus because this is a redox reaction. 4 carbons get converted into 4 carbons, eventually back to oxaloacetate. In this process, we make an ATP, and we make an AFADH2, which is just a molecule which holds an electron, a bit like NAD. The difference is FAD doesn't move, NAD does. So the NAD that's made in the Krebs cycle is going to go off to the next step, which is the electron transport chain.